You could choke down 50 grams of oatmeal fiber every single day and still completely starve your gut microbiome to death inside a month. That metabolic paradox happens because the $40 billion supplement industry sold you a dangerous biological lie based purely on total quantity over botanical architecture. Your lower colon houses over 38 trillion microbial enzymes that cannot survive on a single plant compound, no matter how high the dosage. When you rely strictly on daily psyllium husk or wheat bran, you actively trigger a mass extinction event across your specialized bacterial colonies. If you want the real science on metabolic longevity without the wellness industry hype, hit subscribe and let's get right into the mechanics. To understand why variety trumps volume, imagine your digestive tract as a specialized manufacturing plant with 40,000 unique worker profiles. Each individual strain of gut microbes possesses exact enzymatic keys designed to unlock only one specific type of complex plant carbohydrate. Take Faecalibacterium prausnitzii, our primary producer of butyrate. It completely ignores simple cellulose and demands intricate polyphenols found only in colored berries. Without those exact anthocyanin structures arriving regularly in the lower gut, that vital anti-inflammatory colony simply shuts down its metabolic production lines. Meanwhile, Akkermansia muciniphila requires specialized prebiotic oligosaccharides, like those inside onions and garlic, just to maintain your critical intestinal mucosal barrier. When you dump 30 grams of identical soluble fiber into this ecosystem daily, you aggressively overfeed just two dominant opportunistic bacterial species. This creates a brutal biological monopoly, much like dropping tons of fertilizer on a forest floor and watching weeds choke out ancient oaks. As those overfed generalist strains multiply exponentially, they crowd out rare microbial specialists through a ruthless mechanism called competitive exclusion. The biological tipping point occurs right around 30 distinct plant species consumed every single week, according to global human microbiome sampling. Hitting that 30 plant threshold guarantees your gut receives the exact diversity of microbiota-accessible carbohydrates needed to synthesize thousands of distinct signaling molecules. Those signaling molecules directly enter your portal vein to modulate insulin sensitivity and turn off chronic hepatic gluconeogenesis inside your liver. Yet the average American spends $92 a month on synthetic prebiotics while eating fewer than 8 distinct plant structures all week. Isolated prebiotic powders lack the complex cellular matrix that protects delicate carbohydrate bonds from being destroyed by harsh upper digestive acids. In intact plants, dense cell walls act like microscopic time-release capsules, delivering intact nutrients right to your distal colon where specialists reside. When starved of diverse dietary structures, famished mucosal bacteria switch their metabolic enzymes over to digesting your own protective intestinal lining. That literal cannibalization of your gut barrier lets bacterial endotoxins leak directly into circulation, triggering low-grade systemic inflammation throughout your tissues. This circulating endotoxin cascade blocks mitochondrial energy production in your skeletal muscle, explaining why low-fiber diversity leaves you chronically exhausted. Now, achieving 30 unique plants doesn't mean eating massive quantities. A single teaspoon of 5 mixed culinary herbs counts as 5 distinct structures. However, eating red apples and green apples only gives you one botanical credit because their fiber architecture remains nearly identical at a molecular level. True diversity requires rotating across 6 distinct botanical families: roots, seeds, legumes, tree nuts, aromatic spices, and dark leafy greens every 7 days. Aromatic spices and dark greens carry bound polyphenols that act as chemical beacons, signaling dormant microbial specialists to wake up and multiply. For instance, resistant starch from cooled potatoes feeds butyrate pathways, while insoluble lignins from flax seeds scrub clean your intestinal villi. These diverse inputs trigger a biological relay race called cross-feeding, where the metabolic waste of one bacterium becomes the essential survival fuel for another. Sever just one link in that cross-feeding chain by eating a monotonous diet, and entire upstream bacterial ecosystems collapse within 72 hours. Which raises the critical question: how do you systematically reintroduce 30 botanical structures without triggering severe bloating and gas in an already damaged gut? To understand why high-dose single fibers fail, look inside your colon where over 1,000 distinct bacterial species compete for very specific chemical bonds. Your human digestive tract produces barely 17 enzymes to break down carbohydrates, while your gut microbes possess over 60,000 specialized carbohydrate-active enzymes called sazymes. Think of each sazyme as a custom master key designed to unlock just one exact molecular structure among complex plant cell walls and beta-glucans. When you consume 40 grams of pure psyllium husk daily, you continuously feed the exact same 3 bacterial strains while starving hundreds of beneficial neighbors. Those starving microbes refuse to die quietly. Instead, they activate survival genes that manufacture mucin-degrading enzymes to consume your gut's protective glycoprotein lining. As that 30-micron mucus barrier thins out, bacterial endotoxins called lipopolysaccharides slip directly into your bloodstream and trigger systemic metabolic inflammation within hours. Now contrast that biological erosion with what happens when you introduce 30 distinct plant varieties across a 7-day window, igniting microbial cross-feeding cascades. Primary fermenters like bifidobacteria slice up complex arabinoxylans first, excreting lactate and acetate that immediately fuel secondary specialist strains deeper in your colon mucosa. Those secondary specialists, particularly Faecalibacterium prausnitzii, convert that metabolic byproduct into butyrate, the primary energy source powering your intestinal epithelial cells and tight junctions. Abundant butyrate production signals your enterocytes to consume oxygen rapidly, maintaining an essential anaerobic colonic environment while enhancing peripheral insulin sensitivity throughout your skeletal muscles. To engineer this protective cascade consistently, you must rotate between 4 distinct structural classes of dietary fiber rather than counting total grams on a nutrition label. Class 1 includes highly fermentable soluble oligosaccharides found in leeks, garlic, and dandelion greens, which act as rapid ignition kindling for upper colonic fermentation. Class 2 features viscous, gel-forming fibers inside chia seeds and oats that physically bind bile acids in the small intestine to lower circulating LDL cholesterol naturally. Class 3 involves insoluble structural fibers from celery ribs and kale stems that act like microscopic scaffolding, preventing luminal stasis and speeding transit time. Class 4 comprises resistant starches from cooked and cooled potatoes or green bananas, which survive digestive juices entirely to nourish deep distal colonic strains linked to longevity. Beyond those 4 structural classes, plant polyphenols bound directly to fibrous matrices act as prebiotic signaling molecules that selectively inhibit pathogenic bacteria like Klebsiella. This explains why shoppers spending $6 a box on synthetic fiber bars packed with isolated chicory root fiber still suffer from chronic bloating and insulin resistance. When manufacturers extract one isolated compound and inject it into processed snacks, they strip away the cellular architecture needed to pace fermentation across 5 feet of large intestine. That stripped-down isolate dumps all its fermentable payload instantly in the proximal colon, generating explosive hydrogen gas and osmotic pressure that triggers painful cramping. Intact plant cell walls act like sophisticated time-release capsules, slowly feeding different bacterial colonies mile by mile as digestion progresses toward the rectum. Leading microbiome research confirms that individuals eating 30 different plant varieties weekly show significantly higher microbial alpha diversity than those eating fewer than 10, regardless of total calorie count. That 30 plant target includes far more than basic vegetables. Every distinct herb, spice, seed, nut, and legume brings its own unique phytochemical and fibrous signature. Adding just half a teaspoon of oregano, pumpkin seeds, hemp hearts, and turmeric to your morning eggs instantly delivers 4 complex fiber structures to waiting microbes. However, if your current diet lacks diversity, radically increasing plant varieties overnight will overwhelm your limited seizyme pool and cause severe digestive distress. Your microbial ecosystem requires roughly 14 days to upregulate enzyme production and expand specialist populations capable of digesting newly introduced botanical compounds without excess fermentation. To build enzymatic capacity safely, introduce just 2 new plant species every 3 days while increasing daily fluid intake by at least 16 ounces. Without adequate water, those newly introduced structural fibers absorb luminal moisture like dry sponges, slowing peristalsis and potentially causing mechanical constipation instead of smooth elimination. Furthermore, mechanical prep and thermal cooking dramatically alter how accessible these fibers are to your bacteria, transforming tough, insoluble cellulose into gentle, prebiotic fuel. When you steam carrots or simmer jasmine rice and chill them for 24 hours, crystallization occurs, generating resistant starch structures that bypass small intestine absorption completely. But maximizing your microbiome's short-chain fatty acid output isn't just about what plants you buy. It depends entirely on how you combine and time those specific plant pairings. When you eat diverse plant fibers, primary bacterial degraders break down complex carbohydrates first, triggering a vital metabolic cascade in your gut. Take Bifidobacterium adolescentis, for instance, which specializes in slicing up rigid starch molecules that your human digestive enzymes cannot touch. However, this primary degrader does not produce butyrate directly. Instead, it spits out acetate and lactate as essential metabolic waste products. Here is where the diversity trap springs if you only consume one fiber source. Without secondary bacteria, that acetate just sits there unused. When you consume diverse plant varieties, secondary butyrate producers like Faecalibacterium prausnitzii thrive and rapidly consume that leftover acetate. Think of this microbial interaction like a biological assembly line, where missing one specialized worker halts the entire factory's energy production. That final butyrate molecule is literally the exclusive fuel source for your colonocytes, the specialized cells lining your entire intestinal wall. When your colonocytes burn butyrate, they consume immense amounts of oxygen, keeping the gut lining perfectly hypoxic and oxygen-free. That low oxygen barrier prevents dangerous oxygen-loving pathogens like E. coli and Salmonella from rapidly multiplying and invading your bloodstream. Furthermore, energized colonocytes construct tight junction proteins like claudin and occludin, sealing the microscopic gaps between your intestinal cells. If your dietary fiber variety drops, claudin production plummets immediately, allowing bacterial endotoxins known as LPS to leak directly into your blood. Once inside your bloodstream, those harmful endotoxins trigger chronic low-grade systemic inflammation that blocks insulin receptors across your muscle tissue. Yet there is an even more immediate, destructive danger lurking when your daily meals lack diverse plant carbohydrate structures. Your delicate intestinal wall is guarded by a dense, two-layered protective mucus shield primarily composed of complex glycoproteins called mucins. Specialist microbes like Akkermansia muciniphila constantly prune this outer mucus layer to stimulate fresh mucin secretion from underlying goblet cells. However, if you starve your microbiome of diverse dietary fibers for just 48 hours, starving microbes switch their survival strategy completely. Deprived of external plant polysaccharides, your resident microbes aggressively cannibalize and devour your gut's inner protective mucus lining instead. This self-cannibalization is just like burning your own wooden furniture for emergency heat when you run out of winter firewood. This biological reality explains precisely why swallowing 30 grams of pure psyllium husk daily fails to protect your mucosal barrier. Psyllium provides only a uniform chemical structure, nourishing just a narrow, specialized fraction of your gut's 300 microbial species. Landmark data from the American Gut Project involving over 10,000 citizens revealed the exact dietary threshold for true microbial resilience. Individuals consuming 30 or more distinct plant types weekly exhibited significantly richer, more robust microbial ecosystems than those eating 10 or fewer. Every plant family carries distinct molecular architectures, ranging from pectin in fresh apples to inulin in asparagus and beta-glucans in oats. These intricate structural shapes require specialized bacterial enzymes called carbohydrate-active enzymes, or CAzymes, to unlock their trapped cellular energy. Yet fiber molecules never work in isolation. They arrive tightly bound to colorful plant phytochemicals known as dietary polyphenols. When you eat dark berries or walnuts, 90% of their antioxidant polyphenols pass unabsorbed directly into your large intestine. Once inside the colon, these complex polyphenols act as selective prebiotic fertilizers, suppressing harmful pathogens while feeding beneficial bifidobacteria strains. For example, L-argitannins found in raspberries and pecans are transformed by specialized gut bacteria into a potent postbiotic compound called urolithin A. That resulting urolithin A directly triggers mitophagy, a cellular recycling process that rejuvenates aging mitochondria across your skeletal muscles and brain. Ultimately, every distinct plant color and texture on your plate introduces unique chemical codes that your microbial workforce transforms into systemic longevity. To understand why diversity trumps volume, we must track short-chain fatty acids. Different bacteria ferment distinct plant fibers to produce unique chemical signaling molecules inside your colon. First is butyrate, the primary fuel source for your colon wall cells. Producing this critical anti-inflammatory compound requires specific complex carbohydrates like pectin and inulin from asparagus. When you starve specialist bacteria of those exact plant structures, your butyrate production drops nearly to zero. Without fuel, your intestinal barrier immediately begins losing its protective tightness. Next is propionate, generated when distinct microbes break down beta-glucans from oats and mushrooms. Propionate travels straight to your liver to regulate cholesterol synthesis and glucose release. Then comes acetate, which enters your bloodstream and crosses the blood-brain barrier. There, it directly stimulates your hypothalamus to release natural appetite-suppressing hormones like GLP-1. If you rely on a $20 tub of single-source fiber powder, you might generate some acetate while entirely starving your critical butyrate and propionate factories. That starvation triggers mucus layer cannibalism. Deprived of diverse botanical fibers, famished gut microbes switch on genes that manufacture enzymes to consume your gut's protective mucus lining. Keystone species like Akkermansia normally nibble mucin to stimulate fresh turnover, but without diverse polyphenols and fibers to satisfy them, they aggressively erode this crucial physical barrier. Once that protective mucus thins out, bacterial endotoxins called lipopolysaccharides escape from your gut lumen directly into your underlying arterial blood supply through compromised tight junctions. Within 4 hours of eating a fiber-monotonous meal, circulating endotoxins spike low-grade systemic inflammation. This silent immune alarm actively blocks your cells from properly absorbing blood glucose. To halt this mucosal damage, you need the tipping point discovered by the American Gut Project. Consuming 30 different plant varieties weekly dramatically expands microbial ecosystem resilience. In botanical terms, a plant is not just broccoli or spinach. Every distinct herb, spice, nut, seed, legume, whole grain, and coffee bean counts toward that weekly goal. However, eating 5 varieties of apples only yields minor benefits because they share identical molecular fibers. True metabolic protection requires rotating across completely different biological plant families. The allium family, including garlic, onions, and leeks, provides dense fructooligosaccharides. These exact chains act as targeted rocket fuel for beneficial bifidobacteria residing in your upper colon. Meanwhile, the cruciferous family delivers unique sulfur-containing compounds. When fermented by specialized gut microbes, these convert into sulforaphane, powerful signaling molecules that trigger liver detoxification enzymes. Legumes like black beans and lentils offer galacto-oligosaccharides combined with complex resistant starches. This robust structure survives digestive acids intact to feed distal colon bacterial communities. Resistant starch is particularly transformative. When you cook and cool potatoes or rice, their molecular crystals reorganize into dense matrices that resist human digestion entirely. Those reorganized crystals reach the far end of your large intestine where chronic disease risk is highest. There, slow-fermenting bacteria convert them directly into cellular-repairing butyrate. We must also count polyphenols, the vibrant red and purple pigments in blackberries and pecans. Though technically not fiber, they act as highly selective antimicrobial prebiotics. These polyphenols naturally suppress inflammatory microbes like Clostridium while simultaneously stimulating beneficial species. Think of them as precision weed killers that fertilize only your valuable floral crops. Spending an extra $4 on a multi-seed blend instead of plain chia seeds introduces 5 unique botanical families. This simple rotation dramatically shifts your microbial transcriptome. This brings us to microbial cross-feeding, where one bacterial strain partially digests a tough plant structure and excretes metabolic fragments that nourish an entirely different species downstream. For example, primary degraders break down complex rhi-arabinoxylans into lactate. Secondary bacteria then immediately consume that exact lactate to manufacture butyrate right against your intestinal wall. Without this collaborative cross-feeding network, popping a $50 probiotic capsule containing 20 billion CFU fails completely. Those incoming bacteria quickly starve without diverse physical plant scaffolding. Look at the fiber matrix itself. Intact plant cells release nutrients gradually along 20 feet of intestine, while processed fiber isolates dump everything rapidly inside the upper colon. That rapid, localized dumping triggers explosive gas and painful cramping. Worse still, your downstream bacteria receive zero sustenance because every gram was fermented in the first few inches. Remarkably, diverse microbial fermentation cycles every 12 hours, syncing directly with your master circadian clock. These rhythmic chemical pulses help regulate nighttime melatonin and morning cortisol spikes. Feeding your microbiome identical foods daily freezes those circadian pulses. That internal stagnation disrupts restorative deep sleep architecture and drives intense afternoon sugar cravings through vagus nerve signaling. The good news is the 72-hour adaptation window. Adding just 5 new plant varieties this week measurably alters your active microbial enzyme output within 3 days. Now we need a systematic strategy to scale from 10 to 30 weekly plants without triggering severe bloating or blowing your weekly grocery budget. Most $5 protein bars boast 20 grams of fiber on the label, but rely entirely on single isolated extracts like synthetic polydextrose or chicory root inulin. Flooding your gut with just one isolated compound is like dumping a dump truck of sawdust into a forest and expecting complex wildlife to thrive. That isolated inulin triggers a localized population explosion of just 1 or 2 specialist microbes, while your critical butyrate-producing generalists systematically starve and die off. This microbial imbalance causes rapid gas production and osmotic fluid shifts right inside your small intestine, leaving you painfully bloated without improving insulin sensitivity. To genuinely build metabolic resilience, you must stop tracking simple grams and start counting distinct plant botanical families that trigger multi-layered bacterial cross-feeding. Cross-feeding is a biochemical relay race where primary fermenters break down tough cell walls to produce intermediate acetate and lactate required by secondary microbes. Without those precise molecular intermediates from diverse plant fibers, premier butyrate factories like Faecalibacterium cannot function, regardless of how many oats you swallow daily. Consider the allium family, including garlic, onions, and leeks, which delivers specialized fructooligosaccharides bound to organosulfur compounds that standard grains simply cannot replicate. These sulfur-bound chains specifically fuel protective bifidobacteria colonies in your upper colon, while simultaneously suppressing inflammatory proteobacteria that drive leaky gut syndrome. Next comes the brassica family, where broccoli sprouts and cabbage provide unique glucosinolate fiber matrices that directly upregulate liver detoxification pathways while feeding gut microbes. Your specialized gut bacteria cleave those glucosinolates into sulforaphane, signaling intestinal cells to tighten barrier proteins like zonulin and halting systemic endotoxin absorption. Then consider polyphenol-bound fibers found in wild blueberries and chia seeds, where complex antioxidants physically latch onto tough fibrous carbohydrates during digestion. The fiber acts as a protective armored vehicle, shielding those fragile polyphenols from stomach acid so they reach the distal colon to feed Akkermansia. Once fed by those deep colon polyphenols, Akkermansia accelerates protective mucin turnover and signals your enteroendocrine L cells to release natural GLP-1 hormones for sustained satiety. Don't overlook retrograded resistant starch from cooked and cooled potatoes or lentils, which features tightly packed crystalline structures that human digestive enzymes completely miss. Keystone microbes like Ruminococcus bromii specialize in cracking open those dense crystalline granules, freeing up fermentable carbon fragments for your entire microbial ecosystem. When you eat only wheat bran and oats every day, you select for a monoculture of bacteria that leaves your gut defenseless against sudden dietary shifts. High botanical diversity creates functional redundancy, meaning if antibiotic exposure wipes out one butyrate-producing strain, 5 other specialized species immediately step in to maintain gut integrity. This is why targeting 30 distinct plant species weekly is the true benchmark of digestive longevity, far surpassing any arbitrary gram quota on a nutrition label. You don't need a $100 grocery budget to hit 30 species, because every distinct culinary herb, spice, seed, and tea leaf counts as an independent botanical family. A single teaspoon of dried oregano or ground turmeric introduces complex phytochemical matrices and concentrated polyphenols that rapidly feed neglected bacterial species inside your lower intestine. Yet the biggest mistake people make is doubling their fiber diversity overnight, which overwhelms their existing microbial capacity and triggers severe cramping and abdominal distension. When your gut lacks the required microbial enzyme gene pool to process unfamiliar carbohydrates, opportunistic methanogens seize the excess fuel and pump out trapped methane gas. To prevent bloating, use the 3-day titration method by introducing just one new botanical family every 72 hours, allowing bacterial enzyme production to safely scale up. Simultaneously, you must increase water intake because diverse soluble fibers require hydration to form viscous gels that regulate peristalsis and sweep cellular debris from your colon. Within 21 days of sustained structural diversity, tight junction integrity improves drastically, causing circulating lipopolysaccharide endotoxins in your bloodstream to drop by over 40%. Stopping that endotoxin leakage silences chronic inflammatory signaling in your liver, directly restoring insulin receptors and dropping fasting blood glucose levels without cutting a single calorie. Moreover, the diverse short-chain fatty acids circulating from your enriched microbiome enter adipose tissue to activate uncoupling protein 1, boosting daily baseline calorie burn through thermogenesis. While diverse prebiotic fibers provide the essential construction materials, pairing them with live fermented foods introduces active microbial architects that rapidly colonize and repair damaged gut terrain. And once your diverse gut ecosystem stabilizes, these newly synthesized metabolites travel straight across your blood-brain barrier to fundamentally rewire your dopamine and serotonin production. To put this into action right now, stop eating the exact same 5 vegetables every single day, which only builds a fragile microbial monoculture in your colon. When you skip rotational variety for just 48 hours, specialized fermenters starve and rapidly die off, causing your baseline insulin sensitivity to drop noticeably. Instead of spending $20 on bulk oat bran, make a simple $15 grocery pivot toward unfamiliar root vegetables, seeds, and colorful legumes this week. Dark berries and bitter greens deliver polyphenol-bound fibers that act as chemical keys, unlocking dormant bacterial strains residing deep within your mucosal lining. These newly awakened strains immediately generate butyrate, which directly triggers L cells in your intestinal wall to secrete natural GLP-1 and shut down sugar cravings. If you experience mild bloating during this transition, do not panic or quit. Your microbiome is actively expanding its enzymatic toolkit to process these novel structures. Within 21 days of tracking unique plant species rather than raw fiber grams, fasting glucose stabilizes and those exhausting afternoon energy crashes vanish completely. You can even accelerate this adaptation using culinary herbs and spices like cloves, oregano, and turmeric, which count directly toward your weekly botanical diversity target. Do not waste $50 a month on processed green powders. Dehydrated extracts strip away the complex cellular structure needed to survive harsh stomach acid. The ultimate payoff is incredible. A diverse gut ecosystem boosts metabolic thermogenesis, naturally burning up to 200 extra calories daily without extra treadmill workouts. So stop obsessing over the total fiber grams printed on nutrition labels. Master longevity by counting distinct botanical species in your grocery cart every single week. Hit the like button right now if this science changed your shopping strategy, and comment below with your exact weekly plant count so we can compare. Subscribe to the channel for no fluff metabolic science, and watch this next video right here to see what zero-calorie artificial sweeteners actually do to your gut lining.